Velkommen hertil til vores sommer online møde her, hvor vi har et ekstremt spændende person, der hedder Steve Riley. En praktiker ude i varroatolerance. Lever lige syd for London og har der med hans lokale forening faktisk klaret at have en gruppe biavlere, som ikke har behandlet næsten igennem 8 år, tror jeg. Et par praktiske ting, det er, I kan alle sammen trykke på den, der hedder Q&A. Og der kan I skrive spørgsmål til os under foredraget. Vi vil prøve at notere dem op, og så stiller vi spørgsmålene. Vi har aftalt med Steve, han på nogle tidspunkter vil sige, at så er der tid til spørgsmål, og så samler vi op på dem. Vi har også Ole siddende med her lige nu, som er helt rolig, men der løber vand ind i hans kælder. Så måske henne sidder jeg her lige pludselig fuldstændig alene. Alt efter hvordan situationen er, der er tæt kontakt med Oles kone. Steve, thanks a lot. And this talk will be in English, and I will again go back to Danish. I skulle alle sammen kunne være i stand til at trykke på en knap, således at I får det oversat til dansk. Og det er ikke nogen perfekt oversættelse, men det vil give jer nogle stikord, som I kan bruge til at bedre forstå. Og ellers har Steve nogle rigtig fine slides. Okay, Steve Riley, thanks a lot. Very nice to see you. And we have a time slot until 8 o'clock because Steve is an Englishman. And there is, what was it, a semi-final for the women's European Cup soccer game. And as we know, English people, they cannot live without soccer. And beekeeping, of course. Steve, we know you for a while. We have been watching some of your talks and we find it very exciting. You are the person that we're looking for. Because we have been listening to Stephen Martin. He has been here in Denmark, made some very nice talks, I have to say, very convincing. And now we picked you out or found you because you are the practical beekeeper out there working in your local area, south of London, with the Beekeeper Club. We're very, very excited to see what's going on there. Again, we want to say to everybody, you can make questions in the Q&A. And we pick up the questions and then we will ask them to Steve during this talk. Steve, thanks a lot for being here. The floor is yours. Thanks a lot.
Okay, thank you, Flemming, and thank you, Ola, for organising all the logistics. And good evening, everyone. Apologies for not speaking in Danish. But I have found that most of my Danish friends speak English far better than the English do. So good evening again. We're going to talk about how to transition from non-resistant bees to varroa-resistant bees this evening. Some of the pitfalls of doing so, some of the underlying biology, which Professor Stephen Martin will have talked to you about. But I was going to go through some of that from a beekeeper's perspective. Just by way of introduction, this is us, Western beekeepers. We are sort of southeast of London. Outside the M25, for those who know it, and we have no particular special features to the bees or the area. We have about 130 colonies now, not needing or using miticide treatments, and we're in our 8th season. And the thing that we do is to select for these varroa-resistant traits that researchers unearthed from naturally occurring long-standing varroa-resistant colonies. So we're just copying what nature is doing. Just again for background, in the UK, there's quite a lot of these varroa-resistant traits and resistant bees. And this is from a website which we helped set up with Stephen Martin, that combination of science and beekeepers, varroaresistant.uk, where an awful lot of research for free is on there, and examples and case studies from all of these beekeepers here, which include about 4 or 5 of them are commercial beekeepers. If we add on to that map all of the beekeepers from research back to 2021, which hadn't treated for at least 8 years, you can see how widespread the traits are. And so they're all over the UK. I would think it's the same in most countries. Once you start looking for the traits, you'll start to find them. So today we're going to talk about why we still have a varroa problem. It's over 30 years for us, and I think over 40 years since you had varroa in Denmark. Briefly on the biology of varroa resistance, how to monitor for varroa-resistant traits, how we do it as beekeepers and how everyone can do it. It's not complicated. Some examples of how we've done that. Some thoughts and ideas how to transition to varroa-resistant bees. We call them VRBs. A few lessons learned, things that that caused us some pain that hopefully we can save you some time. And then just to wrap up with how it's gone so far. So why do we still have a varroa problem? Let's just have a look firstly at what varroa susceptible bees look like and how they allow large populations of mites to build up. So if we look at the top left of the screen there, you can see the single mother mite. She's going to try and reproduce in that worker cell, in the red cell there. She goes in there and we're going to assume that she successfully has 2 daughters. So 3 of them emerge from that cell. At the same time, there's going to be a larvae and then a pupa, which is going to be predated on. And in doing that, just in the feeding process, there's little bits of virus particle, which is passed on, vectored into the larvae and pupa and infects that pupa. And this is the problem with mites, that they elevate deformed wing virus. Deformed wing virus has always been there, and it's been in lots of other insects as well, but it's never been elevated in bees until varroa came along. So the next generation, that you've now got 3 mother mites, and they try to reproduce in 3 different cells, and we're assuming that they successfully can do that because there's low defenses to varroa in these bees, and they produce 9 mites in total. And in doing so, 3 more bees are infected. And then by the 3rd generation of reproduction, you have a small army of varroa mites. So these are non-resistant bees. They have low defenses to varroa. They build up large mite populations and with a heavy virus load in your colonies. Now, the bad news is that these are for sale. You can buy these bees. They're for sale all over the UK. And I suspect that's the case in most countries as well. They have low defenses to varroa. They build large populations, as we've just seen, of mites and deformed wing virus. You need to buy and use multiple applications of miticides to keep them alive. And the mites fight back sometimes with resistance, as we've seen, certainly in the US at the moment, new cases. And in some cases, the beekeeper has to wear PPE to protect their own health whilst using these miticide treatments. And these bees cost a lot of money, but you can buy them. You're effectively buying a problem. And we're pushing back quite hard in the UK, and we'll see how that goes. The other problem is that we haven't understood in the past how to select for varroa-resistant traits. And the effect of that, if you start with that little yellow cloud in this cycle, there's no selection for varroa resistance. The bees, therefore, that you produce are susceptible to varroa. They have low defenses to varroa. You need treatments to keep them alive. And once you've kept those bees alive, you then breed from them. So you're breeding from bees with low defenses from varroa. So in this cycle. And at the same time, you never kill all of the mites. You might get 90% of them if you've done well. And in effect, that 10% that survive are the new breeding stock. So in effect, you're breeding for bees with low defenses to varroa and varroa which have very resilient traits. And that's part of the problem of why we still have this issue after 40 years. And if you look at countries like Cuba and South Africa, their whole countries were varroa resistant within 5 to 8 years. Right? And that's with natural selection taking hold there. So you can see how bees have the ability to handle this mite. Is actually the good intentions of beekeepers that have kept the varroa problem going. Here's an example from one of our own apiaries of exactly that. So this is some monitoring that we were doing on mite drop onto the bottom boards through an open mesh floor. And it shows the spring mite drop coming out of winter, the 3 months there. And it's shown on an accumulative basis, so the daily mite drop added up. And you can see along the bottom hives there, hive 4, 3, 6, and 1 are all doing really well. and there's your benefit of monitoring. You can see what your breeder stock is. But Hive 8, I mean, it's doing incredibly badly. This is a colony in the past. We would have done everything to have kept it alive. We would have culled drones. We would have put sugar shakes through it. We'd have used formic acid. And if we had kept it alive, we would have then been breeding from the wrong stock, the stock of bees with very low abilities with varroa. So I think that's a really good example, a real example of how we can get it wrong. But so that Hive 8, in nature it would die, but we've been keeping it alive for far too long. And what you need to do with Hive 8 is to requeen it. But in the green cycle, which is where we are, where you start selecting for varroa resistance, you get bees which are resistant. They don't need treatments. And once you have those bees, you then start spreading. The drones are flying. You're making increases from the right bees, and you just change the whole cycle of varroa. And the other effect on the mites is you have a low level of varroa, and with it a low level of deformed wing virus. So that's effectively rolling back the clock to before when varroa arrived. So there's the wrap up on why we still have a varroa problem. At the heart of all this is a lack of education and training, and so it is solvable. And we've found in the UK two of our three big bee organisations have now come behind varroa resistance. One which is really championing the cause, and the other has now included it in the syllabus if you do bee exams. But there's also activities throughout the whole of Europe as well. So not just the UK, Ireland. I'm on a WhatsApp group there with over 150 people, and it's a crazy WhatsApp group, but there's real fervour to make a difference over there and protect their bees. And there's various breeding efforts in different parts of Scandinavia you may have heard of, in Norway and Sweden, but also across Europe. There's a new big project called Varroaresistente 2033, which there's a big conference at the end of October, early November on that, and the idea is that we will not be using treatments by 2033. So there's a lot of efforts going on just to solve the varroa problem. We know that we can do it now. It's just a question of coordinating our beekeepers. So what's the biology of what's going on? So we see this in three phases for varroa resistance. The first phase is for the bees to find varroa families and parasiticized pupa in worker brood cells. So it's a search phase. The second phase is the removal of parasitized pupa, and in doing so, it prevents the varroa from reproducing. So it stops that big growth that you see in some of these colonies. And the result of that is you get a low level of mites and low level of deformed wing virus. So let's just have a look at that pictorially. So this is a picture we will often see in our colonies, and it tends to be in clusters like this. And it used to be called bold brood. And 10 years ago, bold brood was something which you didn't want to have, and that was what the bee authorities would tell you, and that what you should do is requeen. The mistake we were making is that we didn't understand at the time that this actually was part of the search mechanism for varroa-infested cells by the bees. Now, it usually happens in clusters, but in that cluster, there will only be perhaps one or three of the cells which have the infested and parasitized pupa. And what's happening is that the chemical cues which are coming up through the microporous cell cappings are being wafted around by bees' movement or fanning. And so there's a level of inaccuracy where the bees open up and search some of those cells. So some of those cells will have infested pupa, and I've just for illustration, I've ringed those in red, but the others will be resealed and will just they will hatch out and live a normal life. So the detector bees are the new stars. These have an ability through the antenna, through the mushroom bodies of the brain, and they have this particular olfaction ability, so a good sense of smell for hygienic behavior. And they're nurse bees typically around the age of 11 days old. There's some good work being done by Marla Spivak on that, as I've noted a couple of references at the bottom there, and also Fanny Monday's team in France. Interestingly, the uncapping takes place at the pink to purple-eyed stage. So when you see it, and you will see it in your colonies, I'm sure of it, it will have a look closely or take a picture and blow it up, and you'll see it's the purple-eyed stage, and we'll illustrate why that's important in a minute. Interestingly, there's a cell in the middle there which has been partially uncapped, and you can still see the silk cocoon just in place still. So the second phase, the first is search. The second phase is the removal of the parasiticized pupa, which prevents the varroa reproduction. So you can see here, this is a still from a video. I just saw it happening and managed to get my phone out of the inside pocket of my bee suit. It was in the worst possible place. I was having to undo zips with bees all around me, but you can see there the bees are chewing out the pupa at the purple-eyed stage again. Hopefully you can see that, and they just carried on whilst I was filming there. Now, interestingly, if you have a look at the chart on the right, this is some work done by different bee scientists where they've set up experiments where they've artificially infested brood in different countries, and you can see that varroa-resistant populations will remove infested cells or remove the pupa in about 60 to 80% of the time in the blue, and then much lower levels for treated colonies or those which are not resistant. So there's a real ability that bees have developed, and it's a natural hygienic ability. Here's an example again. If you use your camera and take pictures and blow it up back in the safety of your kitchen, you'll see different things going on. So here's a chewed out bit of pupa, which is they've chewed it halfway down, and I've obviously interrupted them during my inspection. But interesting picture. There's some good bit of science from Fanny Monday's team in France again, which shows that they tested for different chemical cues to see what was getting the bees to do the uncapping, and they used dead pupae, and not surprisingly, almost 90% of cells were uncapped if you just put dead pupae in a cell. But if you just put offspring of varroa in a cell, about 70% of the cells were uncapped, and that was the breakthrough from this bit of research. And just next door, if you just put a single female varroa in a cell, like a mother mite, then the uncapping was at a very low level. So clearly the bees have tuned into offspring of varroa, but also parasitized pupa, which could be a deformed wing virus effect. So let's have a look at this in practice. Okay, so you see the pink to purple eyes and a cell that's uncapped. So what's going on? So this is the 12 days that worker brood is capped. And the main battlefield has to be in worker brood. So worker brood in the UK, and I suspect it's similar-ish in Denmark. Denmark is, we have it from January in very small amounts, and then it's finished by around about November. And whilst varroa prefer to reproduce in drone brood, it's only seasonal. So we'll have that in end of March through to end of June. So if varroa only reproduced in drones, we wouldn't have a varroa problem. So it's the battlefield for varroa resistance has to be in worker brood. That's where they have to solve the problem. So here's the 12 days. You've got the larvae, which has just been sealed right at the start, the top there, and they're spinning its cocoon. Excuse me, and then you can see the pink to purple-eyed stage around days 6 and 7, and then hatching out imago around about day 12. If we overlay the reproduction cycle of the mother mite, so she goes into that cell up to 2 days before, and she hides in the larval food upside down with peritremes just poking up like a snorkel to breathe. Then she's released when the larval food is eaten. And she creates a feed hole on that larvae, and her oogenesis cycle starts, or her egg-laying cycle starts. And she firstly lays a son, a male egg, after about 3 days, and then a series of daughters about a day apart. Now, the blue line there and the magenta lines indicate when those offspring are sexually mature. And it's not until they're sexually mature that they are going to mate. And this is why this interruption at the pink to purple-eyed stage is what we see is incredibly important, in that it happens before the sons and the daughters have been able to mate. So the pupa is removed, the mating doesn't take place, and all of those offspring will die. They're soft-bodied offspring. They're not mature enough to survive. So you stop the growth in mites, and all those existing nymphs and will die. The existing juvenile mites will die. In addition, the mother mite, she usually escapes, but the mother mite has only so many egg-laying opportunities. So she tends to reproduce 2 to 3 times in her lifetime. And this is one life that she's just lost, or reproduction life that she's just lost. And what you find from the research is that in resistant colonies, there's a much higher level of infertile foundress mites, which cannot mate again. So about 45% of them on average are infertile. But then in susceptible colonies, the ones that we can all buy from our bee suppliers, the average is a lot lower, okay, because they're not being intercepted so much. So in effect, there are 3 benefits to this interruption, and this is the really key to varroa resistance. One is there's no reproduction between the son and the daughters, so you get no future population growth. 2, all of the varroa offspring, they die, but the mother mite survives. And 3, the foundress mite is 1 step closer to being infertile. So here's the other half of the quite simple diagram that we talked about at the start of the presentation. And I think just it's useful to see it in contrast. So the varroa resistance, we're looking on the right-hand side of the chart, the varroa resistant bees there. The mother tries to mate, they find what's going on, the bees, and they chew her out. She gets sacrificed, in effect, that bee gets sacrificed. The mother mite escapes, but she goes in again and has another attempt. The same thing happens. And then she tries for a 3rd time, and the same thing happens. And then she's infertile. She's run out of eggs, or she's run out of ability to reproduce. So for the sacrifice of 3 bees and chewing those out, the colony has a low level of mites and a low level of deformed wing virus. And that's what we're seeing in our bees. Right, monitoring and how to get started. So the key thing about monitoring is that this is all about the re-engagement of the beekeeper into thinking about the honeybee and the varroa mite. For decades, all we've been taught and think about is which chemical miticides you use at which time of year. Can you have your supers on or off? Do you need PPE or not? What's the temperature? Which ones work at different times of the year? And we've become expert at that, and we've completely forgotten about this interaction, this interesting, fascinating piece of bee behavior. And it is fascinating. I think people, once you start understanding it, we find it incredibly easy to teach, and people want to understand it. But when you start monitoring, don't just do it once a year. It's a complete waste of time. Start trying to understand and make a judgment on your various colonies of which ones have these abilities and which ones do not. Make sure the approach you use is consistent, so you get some useful data, and that data is going to inform your selection decision. So you obviously want bees with high defenses to varroa, and you want to requeen and get rid of the ones which have the low defenses. We used a varroa insert board. We just went down and kept it as simple as we could, and there were we saw lots of benefits for it. Firstly, you could use it all year. So this is a board underneath an open mesh floor. You can use all year. We'll show you some examples of winter beekeeping. So we don't have to give up beekeeping within the winter. It's non-invasive for the colony. So it takes 30 seconds of just taking it out, having a quick look, perhaps a photograph. It's quick and easy. It's a measure of the mites from the whole colony rather than just a sample. Most of the bee scientists and researchers take a sample of 100 or 300 bees, and they've missed a lot, but that wasn't for us. But the big research win is you started to see the chewed out pupal exoskeleton. So you knew when this hygienic behavior was being undertaken. Helps us understand a bit about the seasonality of hygienic behavior. Again, that hasn't been picked up, I don't think, by the researchers, but you get to see it as a beekeeper and understand it as you follow those bees through the season. But it does need a bit of interpretation, okay? So when there's lots of brood in place, you're going to see initial lower levels of mite drop. The mites that we're looking for are just the mother mites, the dark brown reddy ones, not the juvenile mites you might see. I'm monitoring mine. I have an apiary attached to the back of a garden. Well, if you have one that's within easy access is the best way to get going. You can monitor twice a week, which is currently what I do, just for research purposes. We have other clubs doing it weekly, and our breeding apiary does it weekly. And I have a colleague who was a founder member of our project who just puts her boards in 3 days a month. And I tell her that's not enough, but she tells me she's only lost one colony through the whole of the project and to go away. I think that's a fairly good response. At the same time, you're picking out bits of chewed out exoskeleton. Maybe you can see that on the screen there. There's bits of antennae, which is the key things that you'll see. Just take a picture if your eyesight's not great, and then just blow it up. Here's another example of chewed out pupal exoskeleton all over this board, and sometimes you'll see the bees are really getting on top of their varroa, and we celebrate this. So it's the completion of this interruption of the varroa reproduction, which we like. Keep your monitoring quick and simple. So I just take a little note of the numbers of mites. I also keep an eye on the juvenile mites just in case there's any learnings there. But and then the P is for when I see a pupal exoskeleton. So 9P over 3 days is effectively 3 mites a day, and then a bit of pupal exoskeleton. And I just mark that on and keep an eye on it. We've integrated all of this simply into our hive record cards, into our beekeeping, normal day-to-day beekeeping. This is a free download on the Western beekeeper site, so help yourself if you'd find it useful. So we ask the question, does this queen have the right hygienic traits? Is she a breeder? Do we see the uncapping and the chewing out going on? And then we ask about mites and whether we see uncapping during the inspection time. So very simple to do, and we teach this to our first and second year beekeepers at our training apiary. So here's the wrap up on beekeeper monitoring for varroa resistance. You only need to do 3 things once you've understood the biology. Firstly, look for uncapping, so you know that you have detector bees which can search out for the infested cells. Secondly, monitor for chewed out pupal exoskeleton on the varroa boards. You know that that interrupts varroa reproduction, and the result will be a low level of mites. And with a low level of mites, you get a low level of deformed wing virus. And everyone can do that. Every beekeeper can do that. A couple of examples of case studies, which we'll just quickly run through. So here's an example from my apiary. We have pretty straightforward hive setups, nothing unusual. These are what are called British National boxes, which are a bit smaller than Langstroth. And we just keep them in a single brood box only, which is fantastically easy for beekeeping. They then have a queen excluder. They have their varroa boards in every day, all year round. So in effect, they act as a sort of floor. Bees don't need huge amounts of ventilation. In fact, they really don't like it at all. We use a bit of insulation on the sides of the brood box and on the top of the crown board as well. The bees are locally adapted bees, and they're open mated. They've got darker, interestingly, over time. So we do have the native bee to the UK is Apis mellifera mellifera. And we don't select for it, but the bees have naturally got darker with open mating. We breed from the same queens, their mothers, their daughters, their granddaughters, etc. As soon as you find varroaresistance, just breed from those queen lines. And what we have found is that it gets passed down the queen line. Right? And that was from the early days, where before we started to have an influence over the local drone population, we were finding this. Okay, so it's very simple beekeeping. Just requeen anything which doesn't have the traits with the ones that do, and keep it all within the same apiary. The bees are pretty normal. They try to swarm every year. Most of them do. They tend to produce a good surplus of honey. You know, the big delta, big changes, the British weather, which can never be relied on. And most of the time, they're pretty pleasant to handle. Okay, so they're a good average bee. This is a full year of looking at the hygienic behaviour and the mites from that particular colony. And there's no intervention in terms of what we're doing. There's no treatments, no drone culling. This is just the bees managing their own varroa load. So let's just break that down a bit, because it's quite a complicated chart to follow. So if we just look at we look at the brood here. This is frames with brood on. So during the inspections, how many had a brood? It just gives you an idea of that natural build-up you see in our spring. It might be a month later for you, and slightly different timings, but I suspect the pattern's quite similar. There was an attempted swarm early May, which was an artificial swarm, which we always see as an opportunity to make queen cells, so we have no problems with them trying. And then the new queen starts laying in June very strongly, and then you get that natural fall away as the pollen and nectar starts to reduce into the winter. If we overlay that with the mite drop through the course of the season, the daily mite drop, you start to correlate some interesting findings. So let's think of the start of the year, and we talked about winter beekeeping, winter monitoring. So back in January and February and March, the mite drop's quite low, but it's going lower, and that's partly because of the mites moving back into the brood, but it's still at a pretty low level. So here we are in January, 7th of January of this year, and you can see from the board there, from the wax clippings, you know how big the colony is, across about 6 frames or so. You can see where it is, and you know what the mite drop is. It was it's under 1.5 per month. It's a super colony. The queen has really good varroa-resistant traits in it, and I'm really hoping it gets through the winter. So seeing a good size in January was encouraging. When you look in carefully, you can see there was an egg on the board, and often the queen's a bit over-enthusiastic at this time of year. You will find the occasional egg. So from better monitoring, I know that my queen is laying, so it's a good start to the year. And then we've got the drones being laid in the end of March, early April. And on the boards, you'll see drone caps coming down, so you know what's going on. We know that mites are more successful at breeding in the drone cells. The good news is that relieves the pressure on the workers. So it allows the workers to forage and to build up for their swarming, whilst the drones take the pressure from the mites. You then get into the swarm time, and you can see on the yellow line around May that increase in mite drop. About 170 mites dropped around that time during the period where a lot of the varroa were hatching onto the bees. They had nowhere else to go, and you do tend to see a small pickup during those times. And then the new queen starts laying in June, and lays very strongly up to 11 frames out of the 12 with brood on, and therefore the varroa move back into the brood. So you can see the mite drop is just falling there, and that's why you should never do a once a year mite check at this time of year, because if there's lots of brood there, you'll find that the varroa will be spending most of the time, if they can, trying to reproduce. And then there's this a big spike, which no one had come across before, but so we were doing this for the first time, trying to understand it. So there you are in the sort of late summer into the early autumn with over 600 mites dropping down, which you can imagine when we were doing this for the first time was pretty scary, that we didn't understand it. There was no research on it. And so we had to do our own. So let's just have a look at that, because I think with most people, this would be an emotional moment in their beekeeping monitoring, and they'd be reaching for treatments. Excuse me. So the cause is, a gradual build-up through the drone laying and also through the summer nectar flow. We have a very strong nectar flow in July from blackberry in particular, and everything is subordinated to getting the nectar in at that point in time, because that's the winter stores for the bees. So hygienic behavior is very much subordinated to that. So there's less hygienic behavior during the main summer flow. And then you get this fall away in the brood. You can see the frames of brood going from 8 frames in August down to about 3 in October. And so there's a lot of mites going into less cells. And you get multiple foundress mites going into the same cells. You could have 4 families in there, and these are easy for the detector bees to find. So you see a lot of hygienic behavior going on at this point in time. So in effect, that 661 mites is like a clean out of rower mites as the bees get their nest ready for the winter. And it's, if you got that from putting ApiGuard or Apivar Life on your bees, you'd be quite pleased about it. You'd think that the treatments have done a good job. But this is the bees doing it themselves. The other concept which we learnt about was from a German bee scientist called Torben Schiffer, and he had this concept of store secure, and he had little cameras going into natural nests, and he was able to see hygienic behavior increasing when bees had sufficient stores, which made sense to us. So when bees have got sufficient stores for the winter, they then focus really hard on getting that nest ready for winter. And you typically see that as a beekeeper with all the propolis on your gloves and your hands, and the psychology of the colony has changed. So if you take away all of the honey, for example, you would stress those winter bees. So your winter bees are August to October, and they have to live 6 months. If they're forced to do new work, whether that's processing sugar syrup or having to forage and process nectar into honey for winter stores, then that's going to be a stress on the colony and a reduction of the longevity of that colony as well. So we leave them a whole super of honey on top of the brew box to try and make sure that they know that there's sufficient stores there, and we only take off what we see as a genuine surplus. Let's just have a look in a quantitative way about this 661 mite drop. So this was a colony last October, and you can see they're just starting to reduce the brood laying there. But in that yellow oval shape, there's just over 1,000 cells, and just assuming that there's the same on the other side, you've got just over 2,000 brood cells for brood per frame. And in the example that we've just looked at, there were 5 frames of brood in September, 3 in October, I'm going to assume 1 in November, which gives you around about 20,000 winter bees, or just under. And that could be 15,000 or it could be 23,000, but it's as an estimate. So there's your perspective. That's how many winter bees you have against a mite drop of 661. And that's why you don't need to be emotional about it, but you can think about it in a mathematical way. So under 3% of the mites that came out against those winter bees. And that colony came through into the spring, absolutely doing somersaults, was in good shape. The next part of this chart just needs a little key. So the green P is for white exoskeleton parts, and which you find on the varroa board, and the UN is for uncapping that we've seen during the inspection. And so there's the full year of just the hygienic behavior. So you can see, right, if you go to January on the far left, the hygienic behavior has started already. So you've got all your family out after Christmas, your headache is gone from your New Year's party, and you're doing a bit of January beekeeping, and there it is, hygienic behavior. So 2 bits of good news you've got already is firstly, that your queen was laying about 2 weeks earlier, because fruit of the pink to purple eye stage is about 2 weeks from laying. And the other bit of good news is that your hygienic bees are already taking on the varroa. So the varroa, which have overwintered, will be trying to reproduce, and instead of allowing that big build-up in varroa mites that you'll see from non-resistant colonies, it's not going to happen because the bees are already taking them on. Moving along to June there, this is where the new queen starts laying. She's the daughter of the existing queen. But she doesn't own the colony initially. It takes her a couple of months before those are all her nurse bees. And so if you look at it back to August, you can see that the new queens, there's a lot of green peas there and uncapping. At that stage, the new queen, so the daughter queen, it's her nurse bees that are doing this hygienic behavior. So this was what we started to see, and why we came to believe that this trait is incredibly heritable down the queen line, that you just must keep your daughters and their granddaughters and great-granddaughters, and you'll find that the varroa-resistant traits that you're looking for will be just simply passed on. The only other comment there perhaps is just in the middle of the year, around July, the strongest part of our nectar flow, we do see less hygienic behavior taking place. So this was back to January, again, winter beekeeping. And if you can just see in the middle there, if I blow that up, there's some pupal exoskeleton. So that's and also a bit of antennae there on the top right of that in the purple circle. So in January that hygienic behavior is taking place. And then some other examples of it in a different hive. So this happens in all of our colonies, okay? So it becomes quite the norm. If it's not happening, it tends to be a bit of a worry. So that's so I think I finished this hive off. Let me just move on. So this is we started trying to understand why these daughters have similar resistant traits, why this heritability takes place. And we found some research from John Harbaugh, who was involved in the USDA's project in the United States. And they'd recognized the heritability of this trait. Of course, you only need a small proportion of your workers to be able to do this. So the detector bees, only a fraction of the workforce. So it's not as if the whole colony needs to have all these traits. We do work together on varroa resistance. If you can form a group or with a club, it makes a huge difference passing around the best genetics. And we have tried to influence the drone population, but it's not scientifically proven that the role of the drones yet. Although we think about the simple genetics and believe that drones are incredibly important, but perhaps they're not as important as we first thought when it comes down to the heritability of varroa-resistant traits. That's still to be proven. It's an interesting bit of research, which we've noted along the bottom there, that Stephen Martin carried out on some naive bees up in the north of Scotland, a place called Colonsay in Ireland, which had never seen varroa before, and would have no abilities with varroa. And one of my colleagues in our project, her bees, which are varroa resistant, have been for about 14 years. And what they did was that they put the colonies together and they swapped the queens over. So they put a varroa resistant queen into a colony full of naive bees, and then put the naive queen, never seen varroa before, into the varroa resistant colony. And what happened was very telling. That the colony with the naive queen come down from Scotland, that colony died. So she started laying all her nurse bees, but they didn't have the right abilities. And the queen, which had the varroa resistant genetics, was able to lay the nurse bees and take over that colony, and they were able to control their varroa levels, and that colony got through. So it's a good note. It's on the website that we talked about earlier, the varroaresistent.uk website, as a free download. So what you need is a mated queen with varroa-resistant abilities, and off you go. So there's just a wrap up of that. We talk about this in a bit more detail in the book we published. So just as our framework, everyone has their own ideas of what their mites per day or multipliers should be. So we use 5 a day as across the whole year as our guide. It's never going to be exactly 5 a day, or very rarely. Sometimes it can be nothing or 1. Other days it can be 7 or 10, depending on the hatches and things and the natural flow and workings of that colony. But that to us is a breeder. And then you've got over 10 a day, and for us that's too high. I mean, that's it's over 3,500 mites a year, and those need to be requeened. They're not resistant bees. And then you've got the ones in the middle, which is always difficult, and you have to use your judgment as a beekeeper, as we often have to. So you look at the direction they're going. Is the mite levels getting lower, or are they going up, and take a view on it. This other case study on monitoring is to show you about uncapping and chewing out, okay, and how it actually is a reality, and something we had no idea that was going on in the dark of the hives in between inspection. So here's one of my colonies, same setup as the other one we've looked at, and here's the picture of the same piece of brood comb, worker brood comb, over 2 days, so 24 hours apart. Can you see differences? It helps to have had a photo, because it's we had to get rid of the bees from it, just shook those off. There's a few superstars hanging on there, but let's just mark on the things that have changed in 24 hours. Okay, so in the dark of the hive, this hygienic behavior is happening all the time, all over the place, and we had no understanding of it whatsoever. So the light green coloured cells have been uncapped just in that 24 hours. You've got a bunch of them which have been chewed out in red there, and we just found one that was being recapped, and then you can see that we put it was such a unusual one. We've put a bit of capping, but there's some other ones which have been recapped, which we'll mark on. So there's the activity. If we included the 2 days of action, and an awful lot is happening there. We've included ones which we think have been uncapped and then recapped. Not very clear on the screen, but it was in reality. So an awful lot of activity is going on in these colonies. So some thoughts, okay, about how to transition to VR bees or varroa-resistant bees. But also some health warnings. Be cautious. Okay, so you've got there's an awful lot of non-resistant bees out there, and the people who have been using their own bees will have a better opportunity of finding these adaptations which will have taken place. But just be cautious to start with. Don't just stop treating. Monitor. Be selective. Start looking for the traits. And then just start cautiously with a few of your colonies. I think for new beekeepers, when we look back on our early years, we either got lucky or we had a good mentor if things went well. But your judgment's never that great, and it gets better with some hard lessons in beekeeping life. So just be careful in your early years. Learn the bee craft and enjoy your bees to start with, and then perhaps look at this after a couple of years of experience. And I would say, don't do it with one colony. I think just minimum of 3 to 5 colonies would be a guide. This was our first year. Cautious, very cautious. So we were pretty scared of what was going on. No one was doing this around us. We knew there were varroa resistance happened. There was lots of research on it in different places, different parts of the world. And we'd met a bee scientist called Dr. Ralph Buchler, a German bee scientist at a place called Gormanston in Ireland, and we learned two mechanisms, biotechnical mechanisms of it. One called a queen frame trap, which I just happen to have here. Hang on, get that out. Some of you may have seen this. And the other one was called brood removal, where effectively you just took all of the sealed brood out in August and then replaced it with drawn comb. And both mechanisms work. They replace the summer treatments, and so they did work very well actually. And we found that we knew we never would need to use chemical miticides again. The other thing, really importantly, that we did was not to treat in December. So in effect, the bees and the varroa had 6 months together, okay, right through to the following spring. And then monitoring in that next spring will tell you which of your colonies have abilities and which ones do not. So you remember back to that early slide where the hive 8 had that massive amount of mites coming through. So that's what will happen, and you'll know that those are the colonies that you really don't want going forward that need requeening. Try to use your own queens, your daughters and granddaughters. We've talked a bit about this, and we also try to produce more bees than we need each year. So there's on your top left there, your main honey production colonies. These are evaluated, and we select from the best. We use the artificial swarming time to produce lots of natural queen cells, bee-chosen queen cells, which we like, rather than human decisions on which are going to make the best queens. We make lots of nukes for overwintering, and there's our spare bee populations. These are in poly nukes, so there's good insulation. And then in the spring, if you've had losses, you can replace them with the nukes, or you can sell those nukes into other aspiring beekeepers who want to go down this route. Here's our little incubator. We've got a few of them around the club. Just become self-sufficient in queens. Once you find these varroa-resistant traits, all you want to do is to breed from them and just requeen the rest of your apiaries with them. So these are just cut out from queen cells and very easy to do. Put into roller cages at 35 centigrade, and then you have a small supply of virgin queens running through the course of the year. And these are selected by the bees, okay, which is something that we've found worked incredibly well. This was a club which set up their own project to find varroa-resistant bees. They set up a new apiary, and they brought from all of their club area anywhere they'd found these varroa-resistant traits. So they had the benefit of learning from us, and so they set up this new apiary and just tried to get the best traits there together. They set up right next to us. So Reigate Beekeepers is the club. They're there in the purple, and there's our apiaries in our local area. So they're trying to get the benefit of our traits, but we'll get the benefit of theirs once they've got it right. So they're 3 years in. So that was an interesting way of starting off. Make sure you pass around your best genetic traits. It's an example of one year where we just help each other out with either queen cells or virgin queens or nukes, and just getting people started. We've recently set up a new breeding apiary just to try and increase the number of varroa-resistant queens and nukes that we produce. So before, it's been a bit more of a federal model where the founder members have produced the spare bees, but now we're going to do it a little bit more seriously and try and fill out our whole area. And then we'll be able to help others as well. They get plenty of inquiries from both commercial and non-commercial beekeepers about whether they can have some of our queens. WhatsApp, we've all got far too many WhatsApp groups, but here's just a simple example of one that was set up earlier this year by a beekeeping club called Central Sussex, who are just south of us. They set up a 21-member group and started sharing pictures. All put their boards in the winter. This was in January, remember? And they're on that yellow varroa board there, bits of pupal exoskeleton on the board. So that beekeeper was incredibly excited, shared it with the rest of the group. Everyone thinks this beekeeper is their best friend now. And in the past, you remember, not very long ago, the beekeepers were criticized for not treating, but now they've become the new rock stars. So this lady actually had not treated for 3 years, and then she got her bees from the chair of the club. So already they knew that that there were 2 apiaries which had varroa-resistant bees in them, and off they went. They were able to share that knowledge, and there's a bit of benefit from January beekeeping. One of the ways you can shortcut all this monitoring work and looking for the traits is to collect swarms from free living colonies. So we have bees in trees, not that many of them, but because we've cut most of them down, but also in old buildings as well. So we keep an eye on those. The places where they all have our telephone numbers, and there's a bit of a race to try and get those and be the first. And interestingly, a few of the case studies we mentioned earlier, who have become bee farmers, small bee farmers, actually started off by collecting feral swarms. And one of our members in the Path to Varroa Resistance in the UK group collects swarms from these trees. Joe Ibbison, who took this picture, and he puts the swarms into hives, and he sees exactly the same traits that we've been talking about today. So these traits are naturally occurring. All we're doing as beekeepers is copying what's going on in nature. So there's no making up something new and clever. This is actually just working with the bees. A few lessons learned, okay, which just the pain that caused us will maybe save you a little bit. So like everyone, we all thought that grooming of bees was an important resistant trait. And this has come out of Asia from some research a long time ago, back to 1987. So we started looking at hundreds of mites that have fallen down through the open mesh floor onto the board, and we're cheering away at the damage that was done to them. Bits of the carapace missing, antennae, legs chewed off. Absolutely fantastic news. So we thought we had a real winner there. And this hive in particular, hive 5, had about a quarter of all of the mites were damaged, which we thought was going to be our breeder. And we did actually breed quite a few nukes from this queen. But look at the number of mites there. This is the total mite drop for the whole of 2020. Over 3,000 mites versus others in the same apiary, the same setup of much lower levels. So this was odd to us. We didn't understand why there could be such a high level of mites, but there was a lot of mutilation going on because we'd fallen. We believed the research that was coming out, and this was something we took to the bee scientists, and they did a bit of work on the back of this. Where they reviewed the original research that came out of Asia, which is on Apis cerana bees. And what they found was that the research for grooming that everyone has copied all around the world came about when they'd artificially produced varroa in an Apis mellifera, so a westernized honeybee colony, because they're pretty hard to produce in Apis cerana because they're so resistant against them. And then they put this varroa into the Apis cerana colony. Now, the thing about mites, pretty much all mites have this capability of taking on the smell of their host. So they have a chemical camouflage, right? So they're very hard to smell and in effect seen in the dark. So, but if you take Apis mellifera varroa mites and put them into Apis cerana bees, it wouldn't be a surprise if they got a bit itchy. So they would recognize there was an issue there. So the grooming research wasn't very well founded, and we came to a conclusion that actually grooming and mite mutilation is not a good varroa resistant trait. We do not select for it in any way at all now. So, which was a relief to be able to understand that. We are wary of taking swarms in from unknown sources. We're interested in them from free living sources. So I don't take any swarms at all into my apiary. I just breed from my own, and it's very simple beekeeping and works. A lot of swarms will come from non-resistant bees, so you get choices. If you do find yourself catching them or helping out, which is a good thing to do in the community, you can test for the varroa traits over a few brood cycles, but be aware that the swarms will have a low level of mites, so you won't see them for a while. You could use those bees. You could take the queen out and requeen them with one of your own queens. or you can give them away, put them out of, give them to a new beekeeper, but make sure they're out of your area, not going to influence you in any way. So just be wary of taking in swarms. So just to wrap up with some of results and how we're doing at a high level. This is again a full year of mite drop from 3 different colonies from 2020 through to 2022. So the blue one was one of the earlier ones, and is an example where there were too many mites on the winter bees and too many mites over the year. So there's over 3,000 mites dropped that year, and that's too many. Okay, so that was a colony that was non-resistant. But as our selection processes kicked in, we got better. We got rid of the grooming ideas completely and just selected for lower mites and the chewed out pupa and uncapping. We got better at that, and with some losses of the bees that didn't cope very well. You can see the hive 1 there, which is the one we spent some time looking at. That's a varroa-resistant colony. That's in 2021. And then the magenta line at the bottom, a very, very low level of mites in that colony. And they were really on top of their varroa. And most of our colonies now are somewhere between the yellow and the magenta lines. And we've seen a real stabilizing of our traits, both for varroa and anything else. So it's this is with open mating, remember. We thought we'd better try and see how we did and try to find a good comparator group. So we started off quite cautiously in 2018 and only put some of our colonies into the project, and that was a biotechnical year where we'd used the queen frame trapping and brood removal that we've talked about. That was a good winter for everyone. We compared ourselves, you see in the yellow column there on the right, with the whole of the southeast of England where they are. So that seemed like a fair comparison to us. And most of those will be treated colonies. And if you look down through the survival rates there, you can see that only 1 year were we worse than the southeast. So in total, we're about 6 percentage points better over the season, which surprised us. We thought we were in for a tough time, but actually selecting for these traits, the bees are really good at looking after themselves. And interestingly, this is from our friends up in Northwest Wales. There they have the biggest area of varroa-resistant colonies, probably in Europe, certainly in the UK. There's 500 colonies there now. They haven't been treated for about 16 years. But this was some work they did for 5 years during this project, comparing the treated colonies and versus the non-treated ones in the same area. And you can see there the loss rates were higher for the treated colonies on average over that period of time, and about 6% lower for the non-treated colonies. So I think we're still cautious about drawing this conclusion, but I think we can say that hygienic bees are healthier. They carry less mites and less deformed wing virus, but also that when you put chemical miticides on colonies, it's not a health tonic. It does impact them. And if you read the T's and C's, the terms and conditions in those miticides, it does warn you about that. And I think that's being borne out in the performance of what we're seeing of these varroa-resistant bees. So just to sort of wrap up on transitioning over, start cautiously and make sure you're looking for these key traits, uncapping and chewing out, giving you low levels of mites. If you're seeing uncapping and chewing out, but you've got a high level of mites all the time, then there's insufficient hygienic behavior going on. Work as a group wherever you can and share your best ideas and genetics. Small groups of people who are keen and interested work a lot better. And what you find is that everyone comes into the group who were a bit cautious to start with, but when they see it working, it snowballs very quickly. Raise your own queens. So we talk a lot about locally adapted bees for us, and that's using your own queens. These are queens and bees which are used to your local microclimate, they're used to your local flora, and nectar and pollen flows, and they're used to your local pathogens. They are genetic gold. You're not going to beat that, right? Whatever the marketing is on the bees that get sold. And the other benefit of locally adapted bees is you get this continuity where the adaptation to varroa has taken place. If you're having to buy new queens every 2 or 3 years, you're going to lose that. And the other way to get going is to source varroa-resistant bees from your new best friend who has them. I don't know how popular varroa resistance has been in Denmark. Fleming says not very. Or try to find swarms from free living colonies if you have those. We've written up this in a book called The Honeybee Solution to Varroa, and we were delighted to have a fantastic review from Tom Seeley, who's certainly my hero as I was doing all my bee learning in earlier days. All the royalties for the book are going for bees for development. So we're hoping to spread some learnings on bee behaviors and at the same time do some good. And that's it from me. So I hand back to Fleming, and I'm told there may well be some questions. So thank you very much for listening. Thank you.
And wow, thanks a lot. Really nice, very different, I have to say as well, from our traditional way of thinking into beekeeping. So I found this very, very exciting. There are some questions from the audience, and in respect to the audience, we should ask them first. There's one, really, he was wondering about your insert, and that you didn't paint it white or something like it. For me, he said it was black. For me, it looked kind of wooden colored. Do you have a comment on the insert, how it should be and how it should cover, and what problems there are actually when you're using an insert?
Yeah, so I think the first point is that when I came into beekeeping, all we had was open mesh floors, and that was the norm because we already had varroa here, and that was seen to be a way of reducing your varroa. But actually, bees don't like open mesh floors. And if you find a colony that no one has looked at for 10 years, you'll find they'll have propolized it up. And so and it's the same, bees in trees and other cavities, they don't like having a whole side of their cavity open, and they don't need the ventilation. So having a floor in all the time is a good thing for the bees. They've evolved just to use a small 1 or 2 entrances for all of their oxygen, their CO2, humidity, and temperature management. So don't worry about having it in all of the time. We've used a lot of different ones. The white ones are the worst actually because they're quite difficult to see the pupal exoskeleton. So the pupal, they're good for seeing the dark varroa mites, but not for the pupal exoskeleton. So we've got wooden ones there, which they look quite dark color, but they're just old wooden ones. They have a little lip across the back, which stops airflow and bits flying around. And the white pupal exoskeleton shows up really well against the dark ones. It makes it a bit harder to see the varroa mites, but if you shine a torch or the torch on your phone, the varroa mites, the carapace has a sort of shiny top to it. You actually get quite easy to see, but don't worry about how accurate you are. As long as if you're seeing 8 and it's 10, it's not a problem. Or if you're seeing 6 and it's 8, it's when you're getting it wrong from 70 to 100, there's an issue. So with the small amounts of mites, you're just trying to get an idea of the general quantity, really, and so you don't need to be absolutely accurate about it. So the yellow ones work quite well as well, and that's all we have experience of, really. So initially we started with white, and then we moved over to sort of more natural colours, which were wooden ones with a lip across the back.
Okay, great, thanks. Then maybe there's more a Danish problem or a Danish question, and it's from the same beekeeper. We have aluminium plates that are hollow, that have lots of holes in. So what you could say is kind of, if you have an open mesh floor, there's more hole than there's actually metal. But if you have those, those plates that are kind of, that have lots of holes in, so you could say 50% of the of the bottom board is actually not open. Do you have any experience with that? It's rather common in Denmark. Do you understand what I mean?
I haven't, I'm afraid. Sorry, no, I've not seen those before. Maybe at Appamondia you can show me the. Are they removable? These plates?
No, it's not a plate. It's really, instead of the mesh board, it's a plate that you put on, and then it has maybe 1,000 holes in. That's really the question. Do you have an opinion about that? It's really,
as long as debris can fall through. So the idea of having an open mesh floor, and then a board underneath, is that board is a laboratory for you. That is telling you everything, almost everything you need to know about what's going in that colony without having to open it up. So the more freely that the debris comes down onto the board, the more information you'll have. So that's why the aluminium plate, I can't quite see it, whether it has enough of that openness to allow the debris to come down or not. But you'll have an idea.
Okay, great, thanks. Just to take all the questions, there's a beekeeper asking, do you use drone brood removal at all in your beekeeping, so to say?
No.
So, okay.
Absolutely not. So drones are important, okay, and they take the pressure of the varroa off the worker brood, and they become a pivot to natural selection. So if you have a low level of mites, and those low level of mites go on your drones, then those drones will be more competitive in the competition to mate with virgin queens. But if you have a high level of mites, and those high level of mites will be on your drones, then your drones will be less competitive. So there's natural selection will take place there. So the drones which have a lot of mites will be less healthy, less competitive, less likely to mate, and they won't pass those genes on. So it's incredibly important to allow the drones to do their thing, okay? And at the same time, they are, remember, taking the pressure off the worker brood, which is the key to a successful colony. So we think that's part of old thinking, okay? What we used to get taught 10 years ago, it's still being taught in the UK as well now, but it's part of that red cycle, if you like, that you get stuck in of just breeding the wrong bees.
Okay, thanks. Then we have, I mean, just to tell you about Danish beekeeping, queen breeding is relatively well developed in Denmark. I would even say that we are on the level of supermarket queen breeding, which means I can simply call a queen breeder and I get the queens right away. So they're a rather big production. And now we have a question from a small scale beekeeper. He says, let's say 10 or less colonies. How can he give good feedback to his queen breeder to support him to select the best bees? To decap brood cells. Okay, it says more, a little bit more, and remove mite-infested brood. So how can to put it another way, how can the small beekeeper support his queen breeder to move in the right direction?
Yeah, and I think as us beekeepers become better educated on this subject, we'll demand a different product. Mm-hmm. I mean, the last thing that we want is getting queens and nukes which can't cope with varroa, because in effect, you're just, the bee breeder is passing the problem to the beekeeper. Here's a huge amount of a mite population and deformed wing virus that you need to keep putting miticides on to stop it happening. I think you have to push back and start asking the right questions, which is what we're doing now. We're finding our bee breeders are a little bit defensive about it initially, but some of them are starting to make the right noises about trying to get VSH bees or partly varroa-resistant bees, and so you have to push back and ask the question. So I mean, it's different in every country, and I don't know the Danish the buying habits so well, but in different parts of Europe, you can buy VSH bees. Yeah. And I'm pretty sure they do breed them somewhere. The buckfast bee gets bred somewhere in Denmark on an island with isolated mating. But I would just keep asking for VSH bees. At the very least, they'll be better than non-resistant bees, because you're just you're buying or spending a lot of money to get a problem.
So bees with varroa-sensitive hygienic behavior, is that?
Yeah, VSHBs. And
Yeah,
the bee
exactly.
breeder will know about them already. They all talk to each other, and meet each other at
Yeah.
conventions, and they all understand it. And they just take a view commercially on what they can sell, which is understandable. And they're in business to make a living. But I think, the tide has changed. We now have understanding, and it's time for the bee breeders to come back on board and be thinking about it. And we're not trying to stop our bee breeders from making a living. It's fine. They should be able to make a living, but try to do it in a way that helps all the beekeepers.
Okay, now we have a very practical question. We really have a beekeeper here. He has his insert in his bee colonies. He's counting every second to kind of every 7 days interval regularly. And he sees once in a while a few beige or light brown full-scale cell cappings on the bottom board. And sometimes you even see 20 or more. What do you suggest? What is he seeing there? So it seems that the cell capping is kind of falling down. Yeah. It seems though that the cell capping has been opened up. What is your interpretation of what he's seeing?
It's difficult without seeing it yourself, but yeah. I mean, the color of the cappings is just the color of the wax. So with darker, older wax, you get darker cappings, right? So they just make it out, and with new wax, it's going to be yellowy white cappings. So it could be drone hatches, all hatches at the same. So the queen lays in concentric circles at roughly the same time, then just gradually wider and wider circles. And so there's a lot of hatching at the same time. So it could just be that. It could be uncapping, where bees are investigating. Often you find with uncapping, we saw pictures of it today, but there's a little sort of curve upwards, like a little crater around the outside of each cell, as if they've rolled back the cell capping and they're going to reuse it if they have to recap. But it could be that there's some uncapping going on in the colony. But it's you'll see that at the time of an inspection, you should see uncapping. You should see those little bold faces at the pink to purple-eyed stage, which will give you some clues of whether there's uncapping going on. And if you do that 24-hour picture, it will give you an idea if there's uncapping and recapping going on.
Okay,
so it could be any of those.
So we would kindly ask the speaker, not the speaker, the one who made this question, and I can't see who it is, to send a picture to Ole and me when he sees it, and then please allow us to send a copy of this picture to you if we can't explain it ourselves. Is that okay?
Pleasure, yes.
Now I go up. Now I go up. 1 second. Up here. Yeah, okay, there's one thing. Yeah, if you keep in the bottom board for a very long time during the winter, it will really be filled up with remains, downfalls, dead bees. Yeah, so you have to. So how often do you actually clean your bottom board during the winter?
Every time I look at it. So,
and how often is that?
Well, it depends on what your monitoring period is. So, and it also depends on how easy it is to get to your hive. So I have an apiary in some land off the back of my house. So I do it twice a week. So I always know what's going on. And then I have colleagues who are monitoring weekly. But you could just put the boards in for a month and do it for 4 times over a month, and you'll still give you information about what's happening. But you just need to clean the board each time and then have a look through, see what's going on. There's a lot of information there at a time when you're not opening up the hive. Look inside, and you start getting very good at interpreting what's happening.
Okay, thank you. So there's really no more questions from the audience, but don't worry, I have plenty of questions in my own book here. Could we, I go back to your now we can see your presentation. And could you really go back to that one that says mites per day framework? It's kind of your threshold. And all of okay, there to the threshold. Yeah.
Hang on.
Yeah, and sorry, some of my questions are really a big mess of. I just noted down every time I saw something. Are you back? Okay.
Let me just.
Yeah. Okay. There. No. Yeah.
Mites per day framework. That just goes in a
Yeah.
bit,
Yeah.
I'm afraid. Just bear
The
with me.
threshold one. Yeah. Yeah. Okay. This one. So Ole and I, we have been working with thresholds for many, many years, and we find it very difficult and complicated, actually. And I would say that less than 5 mites per day, to put it another way, 5 mites per day for Denmark is rather high. And if we have a beekeeper getting up to 5 mites per day, we would recommend that one to treat relatively fast. We actually are maybe down on what I call 2 mites per day. What you write here is per year. So I don't know how you calculate those 5 mites. Is it you take all the mite countings and then you divide it with 360, so to say? Is that how?
In effect, yes. So look, there's nothing perfect about this, okay? This is a guide, and that's why we've given you this full year analysis.
Okay.
All right, so that's in the book, for that reason, because we always had this question, and the reality is that mite drop is seasonal, and you start to understand it when you think about it, and take some learnings from other beekeepers. You always see a pickup when there's a broodless period, and there's this pickup, this spike at the end, but that colony there had under 5 mites a day. I can't remember the exact number now, but it's whereabouts was it? Hang on, I've got that here. Let me just show you. We talked about the stability of traits. Yeah. So if we look in the box there, Fleming, that colony in the yellow had 3.5 mites per day. It had 1,261 mites over the year. Right. So it was very resistant. And we've given you the full shape of the year of how it happens and why it happens, and tried to explain it in the book. The sort of key time is actually in this period from sort of early August through to end of October, and because it's the number of bees on your number of mites on your winter bees, which is key to winter survival. And a beekeeper from the Netherlands wrote to me and said all he does is actually monitor mites at that time of the year, and the ones with the lowest mites at that time of year is his breeder queen for the following year. Right. So just to share that, I think where you are is to control the mites at all costs. I don't think you're trying to develop varroa-resistant bees, because varroa-resistant bees around the world, whether it's Cuba, Mexico, UK, Sweden, will have a lot more mites than 2 a day. Okay, and the varroa populations are quite high in these colonies, and it's something that people haven't quite got to grips with. Actually, they need mites to keep bees doing their hygienic behaviour. With no mites, the bees don't react. They don't know how to react. Okay, so the mites are a message. If they're low level and you're seeing the hygienic behaviours being carried out, then you've got bees which are coping well with your varroa. If you're seeing a high level of mites and no hygienic behaviour, then you are bees which are non-resistant. But I think you're probably in a culture of treatment, without having to sort of step back and think, right, how do we get varroa-resistant bees, if you're interested in trying to. And that takes a different culture of thinking, that you will have mites in your colonies, they will be at a certain level, and the colonies, when they come through to the spring and they are absolutely bubbling out of the crown boards and doing really well and obviously very healthy, then those are the colonies which are coping incredibly well with their mite population. So it's you get to a point where you're not paranoid about mites at all. You're quite comfortable with seeing them, and that's where we've got to now. That we rarely see large amounts of mites anymore, but it's good to know that there's a few there, and it's good to know that the bees carry on with their hygienic behavior. So it's a slightly different culture, okay, and it's not one that perhaps you've been taught, and it's one, if you go back 5 years, it's exactly what we had in the UK as well, and it's only in recent years, since this education has been ongoing, that people have started to think differently.
Sorry, can I add a little bit to that, a little side question to this? I mean, but if you're over the season, I mean, when we see these graphs here, it seems that if you could say something, okay, from spring to July or something like that, you should have a stable mite fall below 3-4 mites per day at the most. I mean, don't you need to have something to say, now it's exploding, now it's I have to do something or it will die? I mean, yeah. Shouldn't you have some kind of a threshold or idea to see it's going the wrong direction, I have to do something to save this colony?
Yeah, I think it's quite difficult to do with mite drop. I think you could probably do it with brood analysis. So have you come across the Harbo test? John Harbo was the head of the USDA product. It's free on his Harbo Bees website. And in effect, in August, you're taking a piece of worker brood at the purple-eyed stage, and you are analysing 100 pupae. You're taking them out at the purple-eyed stage to see what the infestation level is. And then they take a view on whether that colony is okay or not. We didn't go down that route because we could not see beekeepers doing that. And you end up just with a few bee breeders doing it, which is what's currently happening in the US. So we've gone down a route of thinking about mite drop, but then thinking about the hygienic behaviors that have to go with it. So do not see mite drop by itself. See it as a result of seeing uncapping, knowing that the bees are looking for varroa infested cells, and see it as a result of seeing pupal exoskeleton on those boards. And then you know that you probably will not have a problem with your bees. So you need to get a bit more comfortable and take a bit more risk, okay, which can be a problem if you're not used to it.
Okay, great. Could you also go to the slide where you have the spring mite drop? Do you know what I mean? This one here, I'm printing while you're talking. It's really, okay. I'm a nasty guy. No, it's just to make my notes.
So this is right at the start. Look away. It's flashing.
Yeah, it's rather early. Maybe, yeah.
It's okay.
There. Yeah, it's a question. So you kind of recommend to count mites relatively often. You do it maybe twice a week or once a week, and it's very exciting. By the way, I do that with 6 colonies at the moment in another project. But to put it another way, I mean, if you take hive number 8, if I would make a counting in the spring and I would make a counting, let's say late May, I would find hive number 8 anyway as one that I should not use. So why should I count mites that often as you recommend?
You don't have to, no.
Because I'm a lazy guy and I want to have varroa tolerant bees as lazy as possible.
Absolutely. Don't count that often, Fleming. No. So just do it twice a year if you like. So do it in the spring and the late summer. There you go. That'll save you 6 months. You don't. So we've done it for research, okay? And different people. I'm at a stage where I really don't need to do it anymore because we know we've got varroa-resistant bees. But the one of the things we haven't shown you is our bee breeding policy, which and one of the points there is how the bees are doing in the spring. So without any treatments at all, how have they overwintered? And you will know in the spring how healthy your bees are just by opening them up. So that colony had quite a lot of brood hive 8, but it also had deformed wing virus bees crawling out the front entrance, and the frames were a mess. And and then the hive count was incredibly high. So it became obvious then, just with that spring monitoring, that there was a problem, and we're able to show that benefit of monitoring because we did the work. Okay, so this was us doing the research originally and sharing with you. So you could just do the work in the autumn, like our friend in the Netherlands was doing, and then you could do it again in the spring, just for say a month, and it would give you some pretty good ideas of which colonies have these abilities or not. But if you're buying non-resistant bees, then you're unlikely to find that you have them. If you're using your own bees and have been for a while, then the adaptation could have taken place.
Okay, thank you. Great. There were one, it's kind of a misunderstanding maybe. It's Thomas, who didn't really understand on the bottom board, but I mean, let me explain and you can correct me. You have a bottom board, it has an open mesh board. It's an open mesh board, and there you have an insert, and then it kind of acts as a closed bottom board, because you say that bees do not like open mesh board all over the winter or something like that. Isn't that right? It's simply asking about this. Yeah,
absolutely. So there's an open mesh floor at the bottom of the hive, which the bees walk across, and underneath that there's a varroa board that goes in underneath that. So there's a small gap between the two, and the idea initially was it would allow varroa to fall down through the colony, was the idea. So before that in the UK, and I think everywhere, we just had solid floors, because a solid floor is really what bees want. And if you look at bees in trees, they have 5 meters above them and 5 meters underneath them, and thick insulation all around them and a small entrance, and that's it. That's what bees want, and it's the same in a lot of cavities as well. And it's the same for people who use bait hives. So they're putting out hives to catch swarms. If you put out a bait hive with an open floor, it catches less swarms, and the ones with a solid floor will catch you more. And bees just look for solid nests rather than ones with where wind can blow in, and it's not natural to them, in the same way that it wouldn't be natural for us beekeepers to live in a house with an open side.
So what they're asking about now in the chat is, what is the size of your mesh? I mean, so that the exoskeleton are coming through? It's kind of a standard one.
About 2.5 to 3 millimeter.
2.5 to 3 millimeters, that's the size.
So we have a professor in Italy doing some work on that. So he's just getting that size of mesh now and starting to have a look at it.
2.5 to 3 millimeters. And then just to finish, the other question is then, so the exoskeletons, they're simply falling through the mesh, and then you look on it under an insert. That's how you're doing it.
Yeah.
Then maybe we are going. Yeah, sorry.
No, that was it. That's as simple as that. So we started to understand about brood removal from the work that was being done 25 years earlier at the USDA. I mean, they had a really fantastic project, and they were showing brood removal taking place, and we thought, well, that makes sense. And then we started seeing because we were counting mites at the time, we started seeing the exoskeleton and started to realize what was going on in our own hives. And this is happening, the same process that we've talked about this evening is happening in hives all over the world, where varroa turned up and there were Apis mellifera bees, so western honeybees, and in different parts of Europe, in Cuba. So there's 240,000 colonies in Cuba that haven't been treated for 20 years. And these are western honeybees in there. You've got Apis mellifera bees with some changes in some of the Caribbean islands, all treatment-free, Mexico, throughout most of South America, South Africa, and most of Africa don't need treatments anymore. It's only the beekeepers and the bee and suppliers who are getting in the way of this adaptation taking place.
May I ask a question about these exoskeletons? I mean, you're saying that you're looking for this, but I mean, is that just you find a little bit, and I mean, do you have like a threshold for that or something? I mean, is it every day you find it, or it's just okay, one day you find it and the next day you don't, or I mean, it's for a beginner in this. What should you look for? Should you just look for it occasionally, or is it something that you find all the time?
Yeah. So most days of the week, but just remember, this is happening at the pink to purple-eyed stage. This is the key moment, the pink to purple-eyed stage. That's when the chewing out process happens to stop the varroa from reproducing. And so it has to be where you have sufficient brood at that age. Okay, so in a new, if after a brood break or a queen has just started laying, she won't have brood at that age initially. So there has to be brood of that age in the colony, and that's when you'll start seeing the chewing out happening. So I just had a quick look down through my hives before we came on here, and all bar one of them had exoskeleton on. Some quite a lot, and some just a little. The other thing that happens is that you get it's quite seasonal. Okay, so in the run up to winter. The autumn time, you'll see a lot more of this going on. And it's as if the bees have really turned on to hygienic behavior and particularly when there's multiple infestation of brood. And that happens after any brood break as well. So in the run up to winter, you see a lot of more hygienic behavior and you'll see it almost every day and sometimes a lot. And at times where the queen has just started laying again, you'll see it. So in the winter, when she starts laying, there's a lot of varroa which are hanging on to the bees and very successfully they do that. And then they go to reproduce and there'll be multiple infestation of cells. And the bees find that the detector bees are onto them. So you see a lot of chewing out at that stage. And then the same time after there's been a brood break, after a natural swarm, or sometimes we have brood breaks when there's a dearth in nectar and the queen goes off lay and starts again. So it happens quite sporadically, but you just get into the pattern of observing it as a beekeeper and getting comfortable with it when you start looking for it. So you'll look through the inspections, you're looking for uncapping. Sometimes you'll see recapping as well, which is where the bees have recapped a healthy pupa and you can see the disturbed surfaces of the cells. I haven't shown that tonight, but it's you can see it if you take a picture of those the cappings and blow it up later. You can see the surfaces where they've been moved over, and then you see then capping. You see the lower mite counts, and you see the pupal exoskeleton, and the three become part of your normal beekeeping observations, and then you come to understand it. But don't expect to see it all of the time. It can happen, it can be every other day, and then it will also depend on what age the brood have in there. So it's just part of your observations and patterns as you go. And the key question you're asking, is there sufficient hygienic behavior going on to give you a low level of mites? And that's the way to think about it. And then you get comfortable with understanding what's happening within that colony. And then you start to rank them. Which of your colonies have better abilities? Which of them have poor abilities with varroa? And you get to know the bees very differently. And it's like if your bees, you know in your apiary which of your bees are mean and defensive and will sting you, and you don't want to breed from those. But you currently don't know which of your colonies have abilities with varroa, which have good hygienic abilities against varroa. And that's what we're going to learn about through going through this.
Great. I mean, slowly we need to get to the end of this meeting. First of all, your soccer game in the basement of Ole. But there are two questions, and maybe this one question is actually too big a question. There's one of our in the audience asking about, did you explain a little bit about your management plan? I mean, how do you extend your bee colonies? Do you use queen excluders? Could you do that in 10 sentences? 10 sentences. How do you run over the year?
We do very normal beekeeping, okay? So we do nothing unusual. And it's the same with our friends in Wales, where they have this huge area of varroa resistance. Normal beekeeping, as if varroa did not exist. They don't even think about varroa. And we're getting there, but we're just doing research and education with it at the moment. So We use queen excluders. We have a brood box, a queen excluder, and then we have a series of honey supers on the top, depending on the nectar flow. And for the winter, we take out the queen excluder and we leave the first honey super on. And that honey super is always the first honey super, and it's always the one that's left on over winter. It's never taken off for honey extraction. So they have a bigger area to lay in. The queen does in the winter, and in the winter we use more insulation. Some of our beekeepers use a lot of insulation in the winter because that's the time you'll get the most stress from the weather, and then the usual things of some people use mice guards. We have green woodpeckers here, so we need wire around the top, keep them off, and then we're back in the spring. In first inspections, we're putting the queen excluder back in underneath between the first super and the queen excluder, and the queen is kept down just into one brood box. We expect them to try and swarm. We're happy with that. We don't like all this idea of trying to stop your bees from swarming. I mean, just work with your bees naturally. And if they want to swarm, do an artificial swarm. We use nearly all of the time one method, and we've all become good at it, called the nuke artificial swarm, which is explained in the book. And then we basically put the queen in the nuke, and with a couple of frames of brood or one of stores, so a 3-frame nuke, which will gradually build to a 6-frame nuke, where you've got your laying queen. Back at the main hive, they will eat you 10 or 20 queen cells. Use those queen cells. If you like that queen and she's got good varroa-resistant traits, use that queen cells, and we put the spare ones in an incubator. So we have a supply of virgin queens from that queen, incredibly easy beekeeping. And then we can use those to make more nukes up, or to help other people start down their varroa-resistant beekeeping journey. And then that's it, really. There's nothing more to it than that. Then we're taking honey off anytime you want. So last year I took honey off early October because we had no pressure on when to take the honey off, because there's no chemical miticides going in those colonies. So you have complete flexibility. I would argue you get more, you have the potential for a higher honey yield, because you can leave honey supers on to be sealed. You can leave them on for longer if you want, or you can take them off whenever you want. So people in our club who treat have to take their honey supers off in August to start their winter treatments in time. But once you have varroa-resistant bees, it's not an issue. If you do we have migratory beekeeping to heather up on the moors, on the highlands, and those people worry about when to treat their bees, but they don't have to with varroa-resistant bees, because it's an ongoing process of the bees just looking for this hygienic activity around the varroa. So that's our full year, really, and it's very simple beekeeping, and the simpler the better. And we just try to work with our bees in a quite an easy and natural way, and we try to fight this idea of sort of the man-made ideas that are thought to be brilliant, and we have this idea that we really look up to commercial beekeepers, and they are fantastic at producing huge amounts of honey and huge amounts of bees and queens, but they're not always doing it in the interests of the honeybee, and that's where we are. So we're a bit more apicentric, and working with the bees, allowing them to help them solve their own problems. We do a bit of selection just to get the varroa-resistant traits in the right place. But after that, it's very straightforward beekeeping.
So now the very last question, and it's actually coming from me as well. In Copenhagen, we have quite a lot of beekeepers there, and we have problems with winter losses because the bee density is very, very, very close. And in your area, how's the bee density there? I mean, reinvasion, isn't that a problem in your club, in your area? Because there must be beekeepers that are not joining you guys in between.
Yeah, we find them, Fleming. We find them. So what you've described to me isn't a problem of reinvasion of mites. The problem is that you've got beekeepers who are mite breeders, who keep bees that are not resistant, which produce huge amounts of mites and huge amounts of deformed wing virus. That's the fundamental problem. But we've got into a defensive mode in beekeeping to try and think it's the mites that are the problem. It's not. It's the bees which do not have these capabilities is the problem. All right. So when varroa first arrived here, the reinvasion was very high from feral colonies in particular, which were all dying and collapsing because they'd never seen varroa in their lives before. But now it's the other way round. The feral colonies actually are quite beneficial. And if you can change the mentality of the beekeepers with some training, education, then we get this. We don't have a problem with reinvasion because of it. So we are just outside of London, all right. What we have found is that we have met so many people now who have resistant bees, who are surrounded by people who treat, and others cannot believe it. And we're at a meeting a club north of London, and we call it the Allison moment. And I was at this club, and they're all being very cynical about this and how it's working. And I said, look, is there anyone here who doesn't treat their bees? And it was a very quiet moment. And then a lady who was sitting right in the dark shadows of the room just put her hand up very slowly and tentatively. And everyone in the audience went, Alison! And they could not believe it. And they couldn't believe the fact that she had bees, and some of them were 400 meters away from other people who had been treating for 10 years. They could not believe it. And that's the turnaround that almost needs to happen. She became the rock star overnight, whereas previously she would have been criticized for not treating. And that's why she was shy about telling people. So it's a little bit about mentality. It's about understanding that the varroa resistance travels down the queen line incredibly well, and that you can do this without other people having the same strategy as you. And we see that all over the UK. People, it's much easier if everyone's on the same strategy. But when we start, let me give you a final example. When we started off with, I think it was 8 of us who started the project. None of us, well, 3 of us were connected, but the rest of us were in different parts of the area, had no connectivity at all. So everyone else around them and around us were treating, right? But we still got the project started. Okay. Every journey starts with a step, okay, and it's about the beekeeper just thinking, hang on, we can do this differently, or the bees can do this differently, and then it's a question of finding those bees. I mean, it really is as simple as that. And when you start looking, you'll see which of your colonies have the traits and which ones don't. And obviously, you know, if you don't have the traits, or you're locked into commercial beekeepers who are producing non-resistant bees, then you'll either have to change them or just carry on and enjoy your beekeeping. No one has to do this, but I think it's in the interest in bees if we can.
Thank you very much. This was a really cool talk, and it might be the start of a new journey in Danish beekeeping. Ole and I already started the journey slowly.
Good luck.
There were some people that asked if Denmark ever will get a project like what you're talking about. We can tell everybody, please write to Ole and me. We do have a project. It's of course it's different, and we are also kind of looking and trying to find where to go and how to go. And you were definitely very, very interesting and very, very exciting for us. What I want to say is this book, everybody has to buy it. It's from a Northern Bee book, and you can see the name up here. What does it say? The Honeybee Solution to Varroa. Steve Riley. Go into the internet and you can buy it there. And probably because, Steve, you're coming to Copenhagen to the Apimonde Congress, so everybody has to come there as well. And then you can go, and then you could do so on Steve's shoulder and tell him, I did listen to you at the online workshop, and then you already have the contact. And Steve, we should have a dinner or something like that. I don't know if Ole and I are under total panic doing the Apimonde Congress, but we would definitely love to invite you and drink some beer or something like that.
It's a good idea.
Yeah. So okay. Thanks a lot. Thanks a lot, everybody. Everybody will get a link as soon as this is uploaded. Ole, when is it uploaded? It's maybe tomorrow. Okay, yeah, great. Thanks a lot. So thank you. See you, everybody.
Okay. Thank you very much for listening. Thank you. Thanks.
Yeah.
Ole.
Bye.
Thank you, Fleming.
Thank you.